I the video. Oh well. We're ready.
My ex-boyfriend keeps sending me pictures where he's just sad. He's just sad. Like, I don't know why he's sending me. Look at this picture. He's just sad. He keeps just sending me pictures where he's sad. I'm like, why are you? Like, what do you mean? It's kind of weird.
That sounds like
It's a weird, like a desperate thing, right? I don't know what he's trying to do.
yeah. He doesn't sound like a fun guy to hang out with right now.
You're so right. Oh my gosh, it's bad. Like, I don't want to just bail like out of his life though. So
Were you friends before you started doing that stuff?
yeah, we're friends. Like we're on good terms and everything. Like after we broke up, but it's just weird. It's just sad.
Sounds pretty nice.
I'm going to go over some of these things for you.
Hey, how many sources did you have for your abstract? Including
Doesn't matter to me.
oh my gosh, were you like running to class? Were you like running to class? Are you okay? Oh my gosh.
From out.
Oh.
Hello, Dr. Preston. How are you today?
Okay.
Yeah.
Oh, we're going straight to the review.
Folks, I am not going to talk over. If you want me to go over this, fine. If you don't, we don't have to.
Okay.
I'm ready for Wednesday. You are.
I'm ready for nothing. Please go over everything.
Okay.
You said it's
Okay.
On photosynthesis. You need to understand about energy, what energy is. You need to know the types of energies, photosynthesis, such as the starts out as solar energy and then winds up being chemical. The chemical energy that photosynthesis converts the solar energy into will be in the form of a very simple sugar, CH2O. And when you add those simple sugars together, you can get things such as glucose. You should know what an autotroph, and this is basic science, things from what, how animals get food. Autotrophs make their own, heterotrophs get theirs from autotrophs. Know the organisms in which photosynthesis occurs that you see listed up there. Those, basically, there's one group up there, two groups that you don't see. What are they?
There's fungi. Sorry. Fungi would be one, I believe.
Mm-hmm.
And animals.
And animals. Fungi are parasites. They're heterotrophic parasites. Algae that we see growing are not fungi. They're in a different group. Algae are protists, as you see here in that group. So the only two groups don't really go through photosynthesis, which are fungi and animals. This gives you some examples. Tells you about heterotrophs and how heterotrophs have to get their energy from autotrophs. Know that chloroplasts are the organelle where that takes place. They are the largest organelle in plants and animals. You need to know the basic structure of the leaves and the chloroplast that you see. So these things that you see here, anytime you see any of these terms that's in bold and black, you should know what they are. Notice that some of these terms can be hard to keep straight, and then when they make sense to you when you read through your notes, but then when you see them on a test, it can get confusing. Mesophyll, chlorophyll, sounds like the same thing, or similar, but chlorophyll is a liquid pigment. Mesophyll are the layer of the leaves that's sandwiched between an outer layer and an interior layer. The mesophyll nose contains 30 to 40 chloroplasts. The pores that open up that allow carbon dioxide and oxygen are the stomata. Thylakoids are the structures that contain the chlorophyll. So make sure you understand and know that. You should know, once again, the equation there, photosynthesis, just like cellular respiration.
Oh, Dr. Preston, will you want that equation or I know that, like when I was in school, I memorized the more simple version, which would be 6 CO2 plus 6 H2O plus, and then, so instead of the 12, do you want the simplified version or do you want this one?
This one,
This one, okay, thank you.
if it had 6H2O, it's wrong. You cannot balance an equation like that.
Oh, I know.
You're going in cellular respiration direction.
So in that last, when I was learning in high school, they didn't have that 6H2O back there. That's what it was.
Water.
Right. Yes, they didn't show that. They only had it up here, so that therefore it was only 6H2O, which was kind of confusing at first, but I see now that it balances out when they put the water there. So actually photosynthesis does produce some water as well? Okay.
It does.
Thank you.
Now, here's the type of things that I want to just kind of, instead of going through these slides, I'm going to want to know what occurs in the light reaction. Anything that occurs in the light reaction you should know, which things such as that is where water will be split, oxygen will be given off, NADP will be reduced to NADPH. That NADPH is going to be sent to the dark reaction. It is linear electron flow. The electrons will go into things such as that primary electron acceptor in photosystem 2, which comes first. The photosystem is at a wavelength of 680. It's called the P680. That will send electrons down the electron transport chain and into photosystem 1, which is at 700 nanometer wavelength. Some ATP will be produced in the process of sending these down, and more NADPH will be generated in photosystem 1, primarily by cyclic electron flow. Paradoxin will be the final acceptor of that electrons that come out, the receptors, or carriers. And then, by way of another electron transport chain, the second one, the energy that has been captured will be sent to the Calvin cycle. In the Calvin cycle, or the dark reaction, carbon fixation will occur. This shows a breakdown of the basic things that happens in the light reaction. The water split, oxygen released, reduced NADP, generates ATP and ADP by photophosphorylation. Calvin cycle is carbon fixation. It is in Calvin cycle, as you see here, it incorporates carbon dioxide into the organic lead. So the big thing about the dark cycle, bring in carbon dioxide, reduce carbon dioxide by adding those hydrogen electrons, protons to it, to where we get then CH2O. That's simple sugar. That's the basic setup. This shows the light reaction and the Calvin cycle. Notice water comes in during the light reactions. Carbon dioxide comes in during the Calvin cycle. And a simple sugar, CH2O, is made. We talked a little bit about wavelengths. You don't have to remember a lot about the wavelengths. I do want you to remember what is the visible light wavelengths. And as you can see, this is the narrow little band right here of visible light, 380 up to 750. Chlorophyll itself, we see green when we see chlorophyll because it reflects these wavelengths that's in this green area, that green band. It's reflected, it's not absorbed, so there's no energy captured from it. It's these other wavelengths where the energy is being captured. Remember, there are more than one kind of pigment. There are two types of chlorophyll, A and B, and they will absorb different wavelengths. But there are other pigments called accessory pigments that we will see. And you should know, this is from lab, and I'm not going to really ask you this, but the spectrophotometer is how we can measure what those wavelengths are. So chlorophyll A is the main pigment. Chlorophyll B is considered an accessory pigment, and some others. Carotenoids, for example, and there are others. There's yellows and different kinds of pigments. You don't have to remember that. And you should understand what's happening when sunlight gets a leak. And if you notice this, it says when the pigment absorbs the light, it goes from the ground state, its normal state, the pigment, these electrons are in a normal state, to becoming excited. Excited simply means they have absorbed energy from the photons. They give off that energy and fall back to their normal ground state. The energy that they've captured and given off is the energy that will be used then to reduce carbon dioxide and to make a sugar, CH2O, out of it. Chlorophyll is an ideal pigment because, as it says there at the end, if you shine lights on it, it will actually fluoresce. It will absorb that energy and capture it for a short period of time before it gives it off. So you need to know what the photosystems are. Remember, the photon comes in, we've got these chlorophyll molecules. There's going to be a special pair of these chlorophyll A. So photosystem 2 is basically working with chlorophyll A. It's primary pigment. It's going to give off that energy. This is what we've already talked about. Notice photosystem 2. Oxygen's given off, water's coming in, it's split. This is the electron transport chain for some ATP is produced. Then we go, that is all linear flow, and then we are going to get into photosystem 1 where it will be cyclic flow. If we get to the end right here, we can see there is a second electron transport chain. And this FD up there is the paradoxin that's transferring that over. And the next step, then all this energy goes into the electron transport chain. So what these primary electron acceptors are, chlorophyll captures the excited electron energy, passes that energy off, goes in to the Calvin cycle. So this primary acceptor is whenever the electron is excited in chlorophyll, it's a way for it to capture it and then move it to where the sugar will be made in the Calvin cycle. Remember, there has to be carbon dioxide come in. That's going to be the base for the sugar. Yeah.
So P680 and P700, I know that there was a difference between the reaction center and the primary electron acceptor. So, okay, so it says P700 accepts, so that would be is that 700 plus? Okay, P700 plus, so that would be the primary electron acceptor, and then P700 would be the,
That would be the one with the electron.
right, P700 would be the reaction center, or P700 plus is the electron acceptor.
Get to where it got both of them up, right here. 700 plus, these will be 700 plus before they get the electron.
Okay.
And that's what that's trying to say up in through there.
Okay.
See, it says P700 X plus accepts an electron, then it becomes the P700.
So we're
Positive
talking.
just simply means it's short an electron.
Okay.
That's why it makes it the carrier.
So when we talk about P700 plus and P700, we're talking about the same thing, but basically in a different state, whether it has the electron or it doesn't.
It's got all of its electrons.
Okay, so but that's referring to one thing, which is that special pair for chlorophyll A, the special
Well, that's talking about
special
a
pair.
lot of them, but these are specialized. Chlorophyll A is the primary. That's the one that gets most of the energy.
Oh.
Some of the other colors, the carrot modes and things will send energy in too. But they said they're there to basically protect the chlorophyll molecule in the leaf because it's at a wavelength of energy, but all the others would be too much. They would be burnout, overload.
Okay.
That's where it was saying in there that they actually protect the cell from those other wavelengths and the whole system.
Oh, thank you.
Notice here it says electrons of NADPH are available. They're sent to the Calvin cycle. The process also removes hydrogen from the stroma that's released whenever the water molecule is split. Notice the NADPH is the electron, right? And this is the hydrogen ion, proton. So not only does the electron go into the Calvin cycle with NADPH carrying the energy, we also have the protons that go. So you can see here, and this just talking about cyclic electron flow. One thing that you should remember, NADPH comes out of photosystem 2. Out of photosystem 1 would be ATP. And what is the difference? NADPH can make about 3. It has enough energy to make about 3 ATPs. Cyclic electron flow is not making enough energy to make NADPH, but it can make one molecule of ATP. In other words, NADPH can carry more energy than one ATP can. Chemiosmosis occurs in a way quite similar to cellular respiration. Same enzyme, notice, ATP synthase. And the same thing happens. We have a hydrogen gradient on the outside of the membrane. That hydrogen proton is pulled back down through, and from that we can make ATP. ATP can then be released and fund the process of making that simple sugar, adding the hydrogen protons and electrons back to the carbon dioxide to make CH2O. This shows what happens again. Once again, it's not that different than what's occurring with cellular respiration, except we're just breaking it down while this is building. This is, if you look right here, this is the stroma. There's the thylakoid. Here's the thylakoid membrane, a membrane just like other cells. Phospholipid membrane with ATP synthase that will send all of these products, the energy, so forth, through the Calvin cycle. Calvin cycle is reducing carbon dioxide to sugar. A question I might ask you is, what is carbon dioxide reduced to? It's reduced to sugar. Where does the energy come from to reduce carbon dioxide into sugar in photosynthesis? If I ask you that question, think, where does the energy come from to reduce carbon dioxide into sugar in photosynthesis? Comes from the light reaction, right? Light reactions produce the energy. Carbon fixation, sugar production occurs in the dark reaction. Notice carbon enters the cycle of CO2 and leaves as a sugar named glyceraldehyde 3-phosphate, G3P. If you take organic chemistry, you're going to find out there's a whole bunch of things out there that are sugars. We think of them as being glucose and dextrose. There's a lot more things that we don't think of as sugars that are like alcohol. And you can actually, you'll see many times some of these sugars actually referred to as an alcohol. So we saw the things that occurred in the light reaction. This is what occurs that you need to remember for the Calvin cycle. 3 phases. Carbon fixation, reduction, what's being reduced again, carbon dioxide, and then it has to regenerate the carbon dioxide acceptor. Carbon dioxide is going to come into the system just like acetyl-CoA came into the citric acid side. What accepted acetyl-CoA, or the acetyl group, was oxaloacetate, and then it made a new compound, citric. Here, what's happening is the carbon dioxide comes in, and what it's carried or accepted by that acceptor, RUBP, which is ribulose bisphosphate. That is catalyzed by the enzyme Rubisco. Rubisco acts as an enzyme. This is the cycle, just quite similar once again to the citric acid, except acetyl group coming in, now it's carbon dioxide, and instead of that acetyl group combining with oxaloacetate, it combines with ribulose bisphosphate. And then we get this, it's called a short-lived intermediate. You got this combination of the enzyme and the carbon dioxide there, and it's starting to get changed into things. And what will it be changed to? 3-phosphoglycerate, bisphospho. You don't have to remember these products right there, like we did in citric acid cycle. Here's what I want you to know about this cycle. Carbon dioxide comes in, it's accepted, it attaches to ribulose bisphosphate, RUBP. It then goes through different stages.
You want to see the 3 phases?
No. Well, these are the, yeah, I want you to know, like, not for this.
Yeah, like, well,
You don't have to go in and point them out where they're at, no. I want you to know the phases, but whenever the intermediate is made, then that carbon dioxide is just going to go through these stages until it is reduced to a sugar. G3P.
Did you say that, so this is one carbon molecule going in at a time, but that has to occur 3 times with 3 different carbon dioxide molecules in order to get one G3P?
Yep.
Okay.
Well, each one will give out, it takes 3 of them to go 3 cycles to get them in.
Okay.
What do you get with one cycle? 1 carbon, CH2O, 2 cycles, 3.
Oh, okay.
You get carbons.
And then you get 3 CH2O, which is the G3P?
You get G3P. Now, there's more than just one of this produced right here. It is saying that one will come out.
After.
That's your output. Some will stay in the system to regenerate RUBP.
Okay.
It is basically the fuel. What's going to happen? You're going to have ATP. Look here, what happens? You've got 1-phosphate, G3, which means the phosphate's on the 3rd carbon, and then you come up here, ribulose. Now it's added 2 carbons, and now it's phosphorylated right in here. It's got a phosphate on each end.
So you said that the carbon dioxide binds to the ribulose bisphosphate, and that's catalyzed by Rubisco.
Yep.
Okay. And then that, when it combines, it's the short-lived intermediate, and then it's turned into 3-phosphoglycerate, which is by catalyst that we cannot
Yeah,
see.
there's a lot of things
Enzyme.
going on in here that they're not talking about. They're just giving an overview of what's happening, and a lot's been left out. All you need to remember, carbon dioxide comes in, Rubisco combines with RUBP, it becomes reduced into glyceraldehyde 3-phosphate that you see right there. Some of that comes out to become a sugar. Some of that will go into regenerate RUBP. And notice it says here, this G3P will be used to make glucose and other organic compounds.
So when does RUBP come in? Is that at the
That is the one that attaches to the carbon dioxide. When the carbon dioxide comes in, this right here combines with CO2 to make that short-lived intermediate. Just like oxaloacetate combined with acetyl group to make citric acid, RUBP combines with carbon dioxide to make that short-lived intermediate that will immediately be turned into 3-phosphoglycerate.
Okay. Wait, RUBP is different than Rubisco, right?
Yes. Rubisco is the catalyst.
Okay.
RUBP is the actual molecule that acts as an acceptor to the carbon dioxide. Don't get too deep into organic chemistry on this, folks. I'm telling you, there's a lot being left out here. Remember what I said. Carbon dioxide comes in, combines with RUBP, catalyzed by Nabisco, is reduced to a sugar, G3P. Some G3P stays in the system and then regenerates RUBP. Now, reducing and the carbon fixation, there are three basic methods depending on the condition, the environmental condition of plants. So you should know those, C3, C4s, and the CAN. We're going to have to move along here or we're going to be out of time. On 11, this is the one, there's not that much I want you to remember, so listen up. Most of the cell communication is discovered in cell biology. There are a few things I want you to remember. Number one, cells have to communicate with each other. That's the way they send messages to tell tissue to move, whatever it is, products to be made. They must communicate. It is normally going to be some kind of chemical message. As you see right there, it says they communicate primarily by chemical messages. And note that signal can be local or distant, depending if it's on local if it's on connecting cells, distant if it's producing a product that goes somewhere else. And that's what these local regulators control communication and function in the local areas. Hormones are the ones that control long distance. And the type of question you might get, what is the long distance signaling mechanism used by plants and animals? Long distance hormones, local distance, local regulators. I do want you to remember these three stages. What happens in the process of signaling? There's three things that happens. One, a signal is received. It's going to be processed. Notice, signal's received, and a signal is then sent. It's processed, and it causes a response inside of the cell. The cell gets the signal, receives it, or the stimulus, it receives a stimulus, and then sends a signal to the inner parts of the cell, wherever it's supposed to go to, and then there will be a cellular response from that. One example is you've got cells in your white blood system, mast cells. If a allergen comes along and stimulates the receptor on that mast cell, that's the reception. Whenever that allergen hits it, it will send a pathway, a signal then to the internal parts of that mast cell, and the response is it will release histamine in the system. And that's what makes your eyes water and all of that. It's the histamine is a molecule that causes usually dilation of vessels. Open up, get bigger. So it'll open up the teardrops in your eyes, and your eyes will water, your nose will run. And it came because you had a signal received, which was the allergen, whatever it is. Then the mast cell sends a message inside to its internal organelles. That's the signal, transduction. And then the response would be the release of histamine.
What does histamine do? Like, why does it be released in response to that?
Why does it what?
Why does histamine get released in response to an allergen?
Because the cell tells it to. That's what we just said.
Well, then
It
what's-
receives the allergen as a stimulus on that receptor, and the receptor then says, get rid of it.
Okay,
And that's what the signal that it's sending is, release histamine, and histamine is a way to get rid of the allergen.
okay.
What it causes is all of that tear flow, runny noses, things like that. And it will also cause blood vessels throughout the body to open up to move things more. I do want you to remember a ligand, the binding between a signal molecule and the receptor. That signaling molecule, that allergen that we were just talking about, is called a ligand. It's a general term for something that stimulates the response. Note also that most all of these receptors are on the plasma membrane of the cell. That's where it's going to come in contact with different things. Do not go into the type of receptors, G proteins, kinases, and all that. That's cell biology. Skip over that. Do not worry about it. The only thing that I want you to remember is sometimes whenever that signal is received, it will release what is called a second messenger. Second messengers just are released throughout that cell by diffusion. There will usually be a receptor. That receptor will go through the membrane. There'll be another little protein, a peripheral protein, in which, when it is stimulated, that sends the second message. I want you to remember, cyclic AMP and calcium are common second messengers. Those will be involved in things such as the muscle contraction and movement. You'll find if you ever take anatomy, there's little compartments called cisternae that are in the muscle cells. It's loaded with calcium, and when it gets that second message come in, it releases that calcium out into the system that will actually make the muscles contract. So that's the same theory, same process, but just a little bit different mechanism. Notice cyclic AMP is very common, second messenger. And notice here what happens. Adenyl cyclase is an enzyme that's in the plasma membrane. And whenever that ligand stimulates the receptor and the receptor starts to want to make that second messenger, adenyl cyclase converts ATP that's present into cyclic AMP, and it does that by basically removing two phosphates. That you'll get, when we go into cell biology, you'll see the importance of calcium. And remember, these, whenever you get that second message, it causes the cell to just release a flood of these ions. And if you're in a big muscle cell and you need a lot of those calcium ions for muscle contraction, that's what is happening. So really, I don't want you to remember anymore at this time about these messengers than what we were just talking about. This other thing, all these different kinds of protein. We did mention apoptosis that you should remember is something that is available for a cell in order to remove itself. Apoptosis is a way of cells recognizing that they are damaged So instead of these cells just dying and releasing what can be very damaging enzymes and other substances out into the system, apoptosis, as it says, is controlled. It's a way of removing that cell in a controlled fashion so it does not release all these damaging enzymes. And that is all that I want you to remember about that chapter. On mitosis, yeah,
Did you say that when the peripheral proteins are stimulated, that's where the second messengers are released from?
that is the second messenger. Yeah.
Okay.
First one is the receptor, and the second one is when the peripheral protein is stimulated that causes the release of these others. They are the second messengers.
Oh.
What they release are the second messengers.
So from the outside, the integral protein is stimulated? I mean, through the...
Receptor on the surface that stimulates the protein on the inside.
Okay, thank you.
Know what cell cycle is? Notice that it has to be there for repair. Note the difference between somatic cells and gametes, what they are. Note that DNA is wrapped in a protein, chromatin, for protection. That becomes chromosomes. So chromosomes are nothing but DNA wrapped in a protective coating. Should know the parts. When you have a chromosome, when it is copied, it will be bound together. Those are called sister chromatids. They will be bound together at a junction called the centromere. When cell division completes, those chromatids are going to be separated. When they separate, then they are called chromosomes. Note the three parts of the cell cycle. You have interphase, mitosis, cytokinesis. Note, chromosomes, the genetic material does not change, it's just copied. This says the cell cycle consists of, these are the same thing that I'm talking about, except they've got it reversed here. Interphase comes first, then comes mitosis, and then comes cytokinesis. That's what I want you to remember. I will ask what are the three parts of the cell cycle. That would be interphase, mitosis, cytokinesis. You need to know what happens in these phases of interphase. Most important thing is S phase is where the DNA is copied. The G1 phase and G2 phase are where the organelles and the cell parts are going to be made. You will need to know these 5 stages of mitosis. Starts with prophase, ends with telophase. The third part of the cell cycle, oftentimes included with mitosis, is cytokinesis, but that is a separate process at the end after the chromosomes have been duplicated and moved to the appropriate places on the poles. Cytokinesis is where they break apart. You need to know about the asters, the mitotic spindles. Remember, the asters are where the spindles attach to the centrosome. Notice up here we've got the centrosome, and then we've got these little things called kinetochores. When you get all of them combined. We've got the aster. Kinetochore microtubules pull the chromosomes apart. Non-kinetochore means it doesn't attach to that centromere. That it pushes against each other to elongate the cell. Non-kinetochore microtubules lengthen the cell. Kinetochore microtubules pull the chromosomes apart. Know what cleavage is in animals where the cells cut off and separate. That does not occur in plants. Instead, when the cell is made, it's the normal size, and the division comes because a cell plate is made, as you can see down on right over here. And these are some of the different stages, what they look like. You guys in the 110 lab will get to look at these. These are in onions. It's what they look like. You can see the faces. One thing to remember. Think of prophase as pretty much being a jumble. Prometaphase is a jumble, but starting to get organized. Notice how they're starting to pair up, getting more organized. And then you see metaphase where they lined up in the middle. Anaphase, they're starting to pull apart, and you can see the little D's as they pull in those directions. And then telophase, over here, you can see, start to see the cell phase, or plate form right there. So you don't really have, in plant cytokinesis, it's the formation of the cell plate. They're not pulling apart. Remember, bacteria multiply simply by binary fission. Just copy and split. And remember, like we were talking about last time, all of these are under control. Cancer cells avoid those controls and grow uncontrollably. This is the cell control that we mentioned last time. Just remember, at these phases, like G1 is one of the most important control phases. What work has been done will be checked at G1, and then if things are not right, then it will not go into the S phase. In other words, the next phase will not take place unless it passes the previous checkpoint. That is a way to make sure all the parts are there and the cells are as they should be. Notice here, these are the checkpoints. You don't have to remember all of them. Just G1 is one of the most important. Usually, if there is a problem that occurs, it will occur in this first part, in the G1. And then, if it does not pass that G1 checkpoint, then it doesn't go further. Notice, for many cells, the G1 seems to be the most important. If the cell receives a go-ahead signal at G1, it will usually do the others and be okay. If it does not get that go-ahead signal at G1, it will exit the cycle, switching to a non-dividing state called the GO phase. In other words, it will shut down that cell's ability to divide and not let it divide if there's something wrong. Kind of like proofreading. And if it's not correct, it will not be allowed to divide. In other words, an unhealthy cell, a bad cell, will not be allowed to divide.
Would that be the same with the cell that the body detects as cancerous?
Well, similar. The problem is the cancer cell has the ability to shut down these checkpoints.
Okay.
So even
So
though
that
it's a
one
bad,
bypasses it.
and even though it's a bad cell, it is able to continue to divide. And then once that bad cell divides, more develop from it, and that's what basically cancer is. Don't worry too much about these regulatory proteins. Once again, that's something we get into in cell cycle. And notice this about cancer cells we were talking last time before we quit. make their own growth factor. They can cause it to grow much faster than normal. They can send their own little hormone to tell the cells to grow even if they don't need to grow. They may convey a growth factor signal without the presence of the growth factor, so they can make it happen, they can fool it. And they have an abnormal cell cycle control system. So, and you can read up on this. This is not totally true right here, but number one, there's no such thing as a benign tumor. All tumors are benign. There is no such thing as a malignant tumor. Cancer is malignant. They can be in a ball shape, but they're not competition, which is more words than anything. But metastasized means spread. Non-malignant growths, such as a tumor, will stay local. They'll do like I told you last time, just ball up and stay in one place. They can continue to grow, but they don't invade other tissue. Cancer does invade other tissue. And this shows like you start with a tumor, they keep getting bigger and bigger. The problem is once these get into blood vessels or lymphatic vessels, then they can be transferred to other parts of the body. It's pretty easy to take out a little tumor like that right there. You can do it with a knife. But when it gets spread to the point that it gets into vessels, you can't do that anymore. So these cancer cells can spread to different parts of the body, wind up in usually the lymph nodes, and start different kinds of cancer in those. And that's why early detection on all of this is always said to be big. Catch them while they're little, small, you can take them out, no big problem. And this shows what you'll see on some of the slides and things, the mitosis. If you look at these, like this right here is going to be going into early telophase. This is anaphase, metaphase, metaphase there and so forth. So that gives you some idea of what we'll be looking at. DUS